Well, good morning, everyone, or good afternoon, good evening, and good night. If you can tell me what movie that's from, then you get nothing. But anyway, so this is the first mini lecture. It's going to be hopefully no longer than 15 minutes, and it will go along with our learning of DNA, RNA, the process of replication of DNA, transcription of RNA, and translation into proteins. So this is part one. In order to understand those other parts, it's definitely important for you to understand these parts. So why don't we just move kind of right into it. DNA and RNA are the fourth major polymer that we have not talked about. The first one is carbohydrates, the second one is proteins, the third one is lipids, and the fourth one is nucleotides, which include DNA and RNA. Well, you've heard a lot about DNA, but we haven't really talked that much about it. What is it? Well, DNA is a chemical storage of genetic information. The information encoded within DNA is information to make proteins and respond to changes. Pretty much every single thing the cell needs in order to function correctly and purposefully is stored in that DNA. Now, some of that information may be expressed constantly, and some of that information will only be expressed once in a while, and oftentimes some of the information in DNA will never be expressed. But the important thing is to understand what the DNA is and its role. Its role is storage information, chemical storage. On the other hand, RNA is a form of nucleotide that differs in a few ways, but RNA is really when DNA wants to make proteins or respond to something in the environment. DNA will, the information of DNA will be made into RNA, and then RNA will make something else. But RNA is a little more diverse of a molecule, and it does more than one thing, whereas DNA just is the storage of information. RNA can be many things. RNA can act as a molecule itself. It doesn't just give rise to proteins and lipids and stuff like that, but generally DNA is the storage of information, where RNA is taking that information and putting it into some sort of form that the cell can use. The DNA is definitely in the nucleus of the cell, as you can see here. A chromosome really is just a really compact form of DNA. All right. So when we talk about chromatin, normal chromatin is in the form of this right here. All right. And then it gets coiled and supercoiled and supercoiled and more supercoiled to produce a chromosome. So when you see a chromosome, what you're really just looking at is a really compact form of DNA. So let's talk a little bit further about what DNA and RNA is. Well, DNA is deoxyribose nucleic acid, all right, and RNA is called ribose nucleic acid. So basically, DNA starts off with a sugar. In this case, it's called deoxyribose because on the second carbon, there's no oxygen. We have a phosphate group attached to the 1, 2, 3, 4, 5 carbon, 5' phosphate. And we also have a nitrogenous base attached. And this nitrogenous base can differ depending on the molecule. So the only thing that really changes within a DNA nucleotide is the nitrogenous base. On the other hand, we go to ribose nucleic acid. Basically the same thing, except it's the ribose sugar, not deoxyribose. As you can see, ribose has a hydroxyl in the number 2 carbon. It still has a phosphate in the number 5 carbon, and it still has a nitrogenous base attached to the number 1 carbon. So ribose is just a sugar that has an oxygen, a hydroxyl group here. And deoxyribose is basically the same thing with no oxygen on the second carbon, making it deoxy, or without oxygen on the second carbon, ribose nucleic acid. So deoxyribose nucleic acid and ribose nucleic acid. The phosphate will always be attached to the 5' carbon, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And this is going to be important later on. And there's always a hydroxyl group sticking out on the number 3 carbon, 1, 2, 3, 1, 2, 3. That's also important to know. So that's basically the chemical structure of DNA and RNA. And these things are the building blocks to the polymers that we know as DNA and RNA polymers. So if we move along a little bit, it's important to know basic differences between DNA and RNA. DNA always exists as a double strand, and RNA can exist as a double strand, but almost 99% of the time consists of one strand, so single-stranded versus double-stranded. On the other hand, DNA is used as a chemical storage of information, where RNA's role is to manifest this information and make it into proteins and other things that the cell can actually use. The cell can't use DNA, but it's like the archive of all the information the cell needs to exist. Whereas if a cell wants to put something into form, it actually takes this information and puts it into RNA form first before we make something else. On the other hand, DNA occurs in the nucleus, where RNA is made in the nucleus, but it migrates out to the cytosol later on. So DNA will never occur anywhere else but the nucleus, and the RNA can be found in the cytosol often. The main sugar in the nucleotide of DNA is deoxyribose, whereas the main sugar in the nucleotide of RNA is ribose. So we have ribose and deoxyribose. The nitrogenous bases, which make up these guys up here, these, like I said, they tend to change. On RNA is cytosine, guanine, adenine, and uracil. So the double-ringed nitrogenous bases are guanine and adenine, and the single-ringed nitrogenous bases are uracil and cytosine in RNA. If we look over here, the only difference is that in DNA, thymine replaces uracil as the single ringed nitrogenous base. Adenine and guanine are the same, and cytosine are the same as well. All right, and we'll see how these kind of pair later on. So keep asking questions. Now, what happens is the nucleotide, which is just one sugar nitrogenous base phosphate conglomeration, actually stack on top of one another to form polymers. Okay, so what do you think? Is this DNA or RNA? If you're saying DNA, you're correct because there's no hydroxyl on the number 2 carbon. What's going to happen here is the sugar and its phosphate, the phosphate group, the 5' phosphate group here is actually going to attach to the 3' hydroxyl of another nucleotide. So if we have this nucleotide here, we have the sugar, and then we have this nitrogenous base. The phosphate sticks out of the 5' carbon, and the phosphate is actually going to bind and form a covalent bond with the hydroxyl group on the number 3 carbon of another nucleotide. On the other hand, continuing, the phosphate group of this nucleotide will form a covalent bond with the hydroxyl of this nucleotide, and it continues like that. Now on one end you're always going to have a phosphate group hanging out, on the other end you're always going to have a hydroxyl group hanging out. Okay. The nitrogenous bases again are the thing that differs. So the sugar phosphate backbone will always stay the same. But the only thing that differs between nucleotide is this nitrogenous base. It could be adenine, it could be guanine, it could be cytosine or uracil or thymine, depending on whether it's DNA or RNA. All right. Now if you look at the other strand here, all right, the other strand, all right, has its 5' phosphate backbone as well. Okay. So very, very important to understand. Now actually, this picture is wrong. So we got to get out of here. I'm going to show you the correct one. Let's see, DNA polymer. That's way wrong. I don't know why I even put that up there. It's good to know. There we go. This one might help us. Remember, RNA is usually a single nucleotide. Get out of there. We don't need you. You stupid wrong picture, you. I don't know how good this is going to be. That's not good at all. Let's get back. This, yeah, let's do this one. This one looks banging. Bam. Okay, let's bring this one. Let's make this one big, just because I feel like it. Okay. So basically, if we're looking at DNA here, we have the sugar phosphate backbone. All right, where the 5' phosphate is always attached covalently to what was the 3' hydroxyl. 5' phosphate, 3' hydroxyl, 5' phosphate, 3'. So we call that, this is one strand of DNA. Now, the other strand, on the other hand, just because of how the nitrogenous bases attach to one another, the other strand is anti-parallel. So it's parallel, but it's going in the other direction. All right? In order for these to bond to one another, nitrogenous bases, the nucleotide or the ribose sugar arrangement on the other strand needs to be different. All right? And that's in order for these to bond. So this is the nature of a double-strand DNA. You have the sugars facing this direction on this strand with the sugars facing this direction on this strand. All right? Now if you notice in the center, the bonds between the nitrogenous bases are all hydrogen bonds, whereas the bonds between the sugar-phosphate backbones are all covalent bonds. So these strands are very strong. The bonds that make the two strands stick to one another are hydrogen bonds. All right? They're not as strong, but remember the DNA strand is thousands and even hundreds of thousands of nitrogenous bases long, of nucleotides long. So it makes sense that the strand is actually kept together pretty well because all these hydrogen bonds kind of interact to form strength between the two strands. But if you notice, the 3' hydroxyl is sticking out here, and the 5' hydroxyl is sticking out here. All right, and that's called polarity. All right, you're always going to have one strand that has a 5' phosphate sticking out, free, not attached to anything, and you always have a 3' on the other side, and the other strand, because remember it's anti-parallel, all right, your 5' phosphate group will be sticking out where the 3' hydroxyl is sticking out on the other strand, and opposite. So anti-parallel, 3', 5'. You're actually going to see later on when we talk about DNA replication, transcription, and translation why this actually matters for the cell, and how the cell kind of interacts with the polarity of the strands to produce. Some of the enzymes can actually only move in certain directions. So knowing this polarity is very important. If we go on to base pairing, remember what I said, the only thing that really differs between one nucleotide and another on DNA is the nitrogenous bases. The nitrogenous bases can be one of 4 things on DNA, can be one of 4 things on RNA. So important is that there are purines and there are pyrimidines, first of all. Purines are double-ringed nitrogenous bases, all right? 1, 2, adenine and guanine, all right? And pyrimidines are single-ringed nitrogenous bases. A double-ringed nitrogenous base always pairs with a single-ringed nitrogenous base. So a purine will always pair with a pyrimidine. Anytime you see a double-ring structure, we'll see a single-ring nitrogenous base that it's attached to, all right? In DNA, adenine always pairs with thymine. All right. So whenever you see an adenine, you'll always see a thymine on the other strand it's attached to. Guanine, on the other hand, will always attach to cytosine. So anytime you see guanine, you will see cytosine. All right. And anytime you see cytosine, you see guanine. And every time you see thymine, you'll see adenine attached. So remember, DNA is always a double-stranded molecule. Okay. So really, the way they fit together are the bonds that make up the carbons and the hydroxyls and hydrogens that attach with the nitrogenous bases. All right, these are what interact, these nitrogenous bases. Okay, so remember that. The important thing is that you know the difference between purines and pyrimidines and how they pair. All right, remember in DNA, adenine always pairs with thymine, and guanine always pairs with cytosine. One of the things about RNA, so if I have a picture, RNA doesn't have thymine. All right. So in RNA, you're always going to see wherever there should have been an A, there's going to be a U. All right. Whenever there's a T, there will be an A, but whenever you see there's an A, you have to replace it with a U with RNA. And we'll talk about that a little bit more when we talk about replication of DNA. So in RNA, there is no A, there is a U. So if we're making RNA and in DNA there's a thymine, we're going to pair that with an adenine, all right, in RNA, because RNA still has adenine, all right. Now, if we're making RNA and we see an adenine on DNA, it will pair not with a thymine, because if we're making RNA, it will pair with a uracil. So it's important to understand. So again, nucleotide polarity This here is DNA. This is obviously RNA because of the hydroxyl on the 2' carbon. But remember, numbering the carbons on the 5' sugar will always be attached on DNA and RNA a phosphate group. On the 5', this is called the 5' phosphate group and the 3' hydroxyl. All right, these are the most important groups for us to know, besides the nitrogenous base. The 3' hydroxyl and the 5' phosphate group. Now, no matter what we're talking about, ribose or deoxyribose, there will always be a 5' phosphate and a 3' hydroxyl. Okay. Again, we're looking at polarity. On each strand, one end will always have a 5' end sticking out and a 3' end sticking out. All right, it's just the nature of how the molecules stack on one another. Okay, 3' end, 5' end. All right. Now, if you were to draw what's going on here, maybe I can draw something. That would be sweet. Maybe. Format, shape. I don't want a shape. I want to straight up draw, man. Maybe. Nope. You probably know something I don't know. Anyway, we won't do that. So polarity is important. If we were to draw a DNA strand, all right, that would join with this one, you know that G would pair with cytosine, good. You know that adenine would pair with thymine, good. And you know that thymine would pair with adenine, all right. If you were to draw the sugars, on the other hand, you would draw the sugars actually would be going in the opposite direction. So instead of the oxygen being facing up on this strand, all the oxygens would be facing downwards. Boom, boom, boom. Okay. Now, remember we said that DNA is the storage of information and that RNA usually manifests that information into something that actually be used by the cell. Well, on DNA, all right, all the information exists in pairs of 3. All right, so these 3 nitrogenous base pairs, like for this one, 1, 2, 3. These 3 nitrogenous bases would be information for something. All right, and they would be read TAG or GAT. All right, but this pair of 3 is going to actually code for a specific amino acid way later on. So if this coding was TAG, all right, well actually we read 3 to 5. Sorry, and that's important to remember for now. We read 3 to 5, so it's GAT. All right, and I'll tell you why later. Let's go here and see what GAT would make. Well, if we went to G, A, all right, and remember in RNA T is replaced with U, so we're looking at GAU. GAU makes aspartic acid. So that would be the amino acid that would be made from that codon sequence. It might be a little bit fuzzy for you right now as far as what the codons mean stuff, but just kind of giving you a warm-up as to what we might be seeing later.